In this first lab practice, we extracted plant DNA from a polio-grinded tissue by a variation of the CTAB protocol modified for the extraction from aromatic plants. To achieve the extraction, we use some reactants such as extraction buffer, lysis buffer, chloroform, isoamyl alcohol 24:1, 3 molar sodium chloride, 5 molar sodium acetate, ethanol 70% and 95%, tea buffer, and cold isopropanol. We also use equipment such as the centrifuge, fridge, and water bath. After several hours of lab work and some mistakes, we extracted the polio DNA. The practice took a little longer than we expected because of our lack of pipetting experience and the time that all the centrifugations took. Knowing the function of each reactant and equipment, it's a very important step to do, and we have to get used to it. That way, it's easier to modify the protocols depending on the needs and changes of each practice.
As biotechnologists, one of the basic techniques that we have to manage is extraction of DNA. There are many ways to perform the protocol, although the reactants have the same use. For example, extraction buffer, EDTA, that is an chelating agent. After being bound by EDTA into a methyl complex, methyl ions remain in solution but exhibit diminished reactivity. And chloroform, isoamyl alcohol. Each chemical is used to help separate proteins and polysaccharides from nucleic acid. Chloroform is more dense than water solutions, thus after centrifuge solutions, the chloroform will separate from the water. The lower phase will be chloroform alcohol, proteins, and polysaccharides. The upper phase will contain the DNA. With plant tissue, there are often more polysaccharides than in animal tissue.
The lysis buffer that we are going to use depends on the type of nucleic acids, such as genomic, mitochondrial, or plasmid DNA, and total or a subtraction of RNA as well as the cell source. This solution is used to breaking open the cells. Also, isopropanol, pure ethanol, and salts are used to precipitate DNA from this solution. Finally, ethanol at 70% is used to wash the DNA and remove the excess of salts.
As first step, we weighed out 20 milligrams of dried plant tissue that has been grinded before. After that, we have to add 600 microliters of extraction buffer. And mix the solution with a vortex twice for 8 seconds. Before the centrifuge step, we have to make sure that the two samples weighted the same. So we weigh and calibrate and then centrifuge at 4 degrees for 10 minutes at 11,600 RPM. The next step is remove the supernatant and add 400 microliters of lysis buffer. Then we have to mix the solution with a vortex again. Then we have to incubate the samples at 65 degrees for 30 minutes, making sure that we mix the solution every 10 minutes. After that time, we have to transfer the samples to ice and add 500 of chloroform isoamylic alcohol and mix well by immersion. Again, we have to weigh the samples and centrifuge at 4 degrees for 10 minutes at 11,600 rpm. After the centrifugation, we should have 3 layers. In the top, the aqueous phase, in the middle, the debris and proteins, and in the bottom, the chloroform. These phases shouldn't remix, so we have to go to the next step so quickly. Pipette off the aqueous phase, taking care not to suck off any of the middle of chloroform phases, and place the aqueous phase into a new labeled plastic tube.
Estimate the volume of the aqueous phase, measure the new volume, and add the same quantity of 24:1 chloroform isoamyl alcohol as the recovered volume of the supernatant.
Centrifuge at 4 degrees Celsius for 5 minutes at 8,700 rpm. Place the aqueous phase into a new labeled plastic tube. Measure this new volume. Add the same quantity of isopropanol as the recovered volume of supernatant. Add 10 microliters of 5 molar sodium chloride mixed by immersion.
Centrifuge at 4 Celsius for 5 minutes at 8,500 rpm. Pour our pipette of the liquid, being careful not to lose the pellet with your DNA. Resuspend the pellet in 100 microliters of TE buffer plus 10 microliters of 3 molar plus 1 microliter of RNase. Incubate samples at 37 degrees for 30 minutes.
Place samples on ice and add 3 volumes of absolute ethanol. Centrifuge at 4 Celsius degrees for 3 minutes at 8,700 rpm. Pipette off the supernatant and add 1,000 microliters of TE buffer plus 10 microliters of 3 molar sodium acetate plus 3 volumes of absolute ethanol. Centrifuge at 4 Celsius degrees for 3 minutes at 8,700 rpm. Pipette off the supernatant and add 3 volumes of ethanol at 70%. Centrifuge at 4 Celsius degrees for 3 minutes at 8,700 rpm. Pipette off the liquid, being careful not to lose the pellet with your DNA.
Dry the pellet and place the samples in the spit vac. Resuspend samples with 100 microliters of TE buffer and allow to resuspend for 1 hour at 55 Celsius degrees or overnight in refrigerator. In this practice, we learned something very important for our career, that is the DNA extraction, which is the basis of biotechnologies. And we also learned the method and how to use the equipment and what are the substances for. So we think that was very useful for us.